So now we have cell division and reproduction part 3. So, meiosis. I think I shall call him Mini-Me. Because meiosis is making the sex cells to make babies. So, starting off with chromosome number. Organisms have many genes that determine individual traits. Genes are located on chromosomes. The homologous chromosomes are look-alike pairs of chromosomes. Each person has 2 copies of each chromosome. So an example, you may have a 21st chromosome over here, and then you'll have another 21st chromosome over here. You have 23 homologous chromosomes. You get 1 set from mom and 1 set from dad. So 23 and 23, you get 46. Again, looking at homologous chromosomes. I have a number 1 from mom, and I have a number 1 from dad. These 2 together are homologous chromosomes. They're look-alike. This gene right here on both of them may be hair color. This gene right here may be eye color. Now, that does not mean that the chromosome you got from mom has to be the same hair color as the one you got from dad. They can be different, but they encode for the same thing. Talking about chromosome number, we also talk in ploid, or in sets of chromosomes. Diploid, which is given as 2N, is when a cell has 2 of each kind of chromosome. So in humans, we have 23 pairs, or 46 chromosomes. So we are known as diploid. In some cells, they are called haploid, or haft, which is known as N, small n. It's a cell with 1 of each kind of chromosome. So in the human egg and the human sperm, they are haploid, and they only have 23 chromosomes. That is so. When the egg and sperm combine, we can have a zygote that has the correct 46. Meiosis. So the phases of meiosis, the process of 2 divisions where the chromosomes are cut in half. So we have characteristics, the process of sexual reproduction to create the gametes. We create 4 gametes per meiosis. And they are haploid, only having 23 chromosomes. That's half the normal amount, half the diploid amount. And they are known as the sex cells. And they are genetically different. They result in half the genetic material, and they go through PMAT twice. Interphase is the same, and it starts out in front of meiosis. So again, we have G1, we have S, where we're doubling our DNA and making more organelles, and then we have G2, which is preparing for the division. And then we go through meiosis, which is PMAT twice. So again, interphase, then we have prophase, metaphase, anaphase, telophase. The way that you can know that it's prophase 1 or metaphase 1 is you're looking at one cell. Then afterwards, after telophase, you're looking at prophase 2, metaphase 2, anaphase 2, and telophase 2. You know that you're talking about prophase 2 and metaphase 2 because you're actually looking at 2 cells. So in meiosis 1, specifically prophase 1, we have a few things that are different. We have synapsis occurs, which is the physical pairing up of those 2 homologous chromosomes. So they're side by side and form a tetrad. Tetra means 4. So if we look here, that's 1, 2, 3, and 4. Paternal is father, maternal is mother. So these are the mothers, and this is the fathers, and this forms a tetrad. In this tetrad, they sometimes touch, which then crossing over can occur. Now, DNA is sticky, so if it accidentally touches, when it pulls apart, they may swap genetic information. This is right here. Now, this paternal has part of mom, and part of mom has part of dad. This increases genetic variation. In metaphase 1, again they line up in the middle, but they line up as tetrads. We see a double file line. So here we have the tetrad, and they're lined up in the middle. spindle fibers still connect to those centromeres. In anaphase, instead of the sister chromatid splitting, we have the actual homologous chromosomes are actually going to either side, resulting in dyads, or a chromosome consisting of two sister chromatids. This adds diversity via independent segregation, which means it depends on how the pairs line up. Moms don't all line up on one side and dads on the other. Sometimes they're just completely mixed. So depending on which chromosome you get, whether you get mom's number 1 and dad's number 2, that can add variation. In anaphase, again, these chromosomes with two sister chromatids are pulled apart. Telophase is the same as mitosis, so the opposite of prophase, which is CN/CS, and it results in two cells, and they go straight into meiosis II. They do not hit interphase. No interphase. Go straight into meiosis II, which is prophase II, which is dyads, which are composed of the pair of sister chromatids. We see CN/CS, looks remarkably like mitosis prophase. Then we have metaphase, where the centromeres line up in the middle. Looks identical to mitosis, except there are two cells that we are looking at. Anaphase, again, looks identical to mitosis. The sister chromatids are pulled apart, resulting in monads, or one sister chromatid on either side. Again, looks identical to mitosis. And then we have telophase. Again, the same as mitosis, except it's resulting in 4 haploid cells. Remember, haploid means that there's only one set of 23 chromosomes. These are going to be our sex cells, or gametes, which are our sperm and egg, and they are genetically unique. So, meiosis II, we're talking about 2 cells, 2 cells, 2 cells, and then we end up with 4 cells. And these look, prophase II, metaphase II, anaphase II, look almost identical to mitosis, except at the end we get 4 cells instead of just 2. So when we're talking about meiosis of sperm cells, we end up with actually 4 sperm cells. For an egg, which houses all the organelles and the things that the cell will need, we actually create 1 egg cell and 3 polar bodies. Now these polar bodies are discarded, and they actually break down and provide nutrients and supplies back to the organism. So we get 4 sperms, but we only get 1 egg. And that egg contains all of the organelles needed to start the actual zygote once the sperm comes in. Therefore, we can actually have division and see a baby form. Comparing mitosis and meiosis. Again, mitosis can occur in body cells. It's mostly for repair and growth. It's not for the sex cells. Mitosis produces 2 genetically identical diploid cells. And we have asexual reproduction. Meiosis, 4 genetically haploid gametes, the sex cells. And this is for sexual reproduction. Genetic variation occurs in meiosis so that we can be unique. We have genetic recombination, which reassorts the chromosomes either by crossing over or independent segregation of the homologous chromosomes. These increase genetic variation. Well, that's it. Bye.